So the title of my PhD was Quantifying the Response of Macroinvertebrates to Gradients of Fine Sediment Pollution. And I'm going to try in the next 30-40 minutes to give an overview of the whole of my PhD, so the different methods that I use, and also a bit of an insight into my postdoc and what I'll be doing over the next year. So let's get started. Firstly, what's the problem? Less than a quarter of our rivers in England are considered healthy and determined by the Water Framework Directive, which is part of our international obligations that we are legally bound to report to. And this figure has continued to fall since we first started monitoring within that framework over 15 years ago. A primary cause of this decline is fine sediment, and what I mean by fine sediment in the context of my PhD is sand, silt and particles smaller than 2 millimetres. And there's a variety of different sources: agricultural land, construction, mining, deforestation, anything really that destabilises the upper layers of the soil, which can increase soil erosion and runoff into rivers. But there's also in-channel sources as well, which is a result of how we've over-engineered our river environments over the years. But within this country, trace studies for up to 70% of the fine sediment which ends up in our rivers. So what happens when it gets to river environments? It clogs the riverbeds, reducing oxygen exchange. It buries and smothers organisms that live there. It creates a homogeneous habitat with very low diversity. It blocks light, affecting primary production and having other knock-on effects on the trophic web. It can also transport harmful chemicals. So when we think about agricultural sediment runoff, this could be insecticides, pesticides, or nitrates and phosphates. And it also impounds river channels. So this can have an impact on migratory species, but also has requirements. So as well as having significant environmental impacts within river environments, it also has a significant effect on land, and soil degradation in England, Wales costs farmers and wider society an estimated £1.2 billion per year. And because of this and recognition of the environmental effects, there's been a huge monitoring drive, because with better monitoring practices, we can more efficiently identify areas requiring land management interventions to try and help ameliorate the problem. This brings me on to my PhD project aims. I'm kind of presenting in 3 key phases today, which is just how I'm sort of structuring my aims and my results. So my first phase was to review what we know about macroinvertebrate responses to fine sediment. My second phase was to try and improve our mechanistic understanding of these effects, and my last phase was to test the response of macroinvertebrates to measurements of fine sediment. Starting with my I'm showing here essentially a complex mix of direct and indirect effects through which our pressure and increase in fine sediment can affect and change community composition. When we talk about fine sediment as a pressure, we often divide this into the 2 main fractions: deposited sediment and suspended sediment. And in order to properly monitor the effects of this, we need to understand exactly how this is changing and affecting community composition. So this was the key focus of my review. There's many different methods and evidence review at our disposal, and they span a gradient of time, cost, critical appraisal, all the way from sort of basic literature reviews to full-scale meta-analysis. And traditional literature reviews are a very useful tool for synthesizing evidence. However, they can be prone to bias. And there already are existing literature reviews, fine sediments on a variety of different taxa. So I didn't want to repeat what had already been done before. Reviews which are more systematic within their methods follow a defined protocol. They can help answer more specific research questions, and you can also apply a weight of evidence approach, so looking at evidence quality as opposed to just quantity. The method that I decided to use was a blended approach of systematic mapping, so I could look at the breadth of evidence and also incorporating a rapid evidence assessment. So looking, that's where I was looking at evidence quality as well. My first phase was to carry out my evidence search. So I searched 2 databases with a very broad search term, which is why I had so many articles. The N represents the number of articles. And then I underwent a series of screening phases. So my first phase, I looked at the article title. My second phase, I looked at the abstract, and then I did a full text article assessment for eligibility. And I ended up with 121 different papers. And then each paper can yield a single line of evidence of macroinvertebrate response or multiple lines. I then needed to extract evidence from each publication. So I'm defining this within 3 categories. So I extracted evidence, information about the predictors, which was the type of study, and then a bit of information about how fine sediment was quantified and the methods that they used. The responses were the actual response of macroinvertebrates and how they define that. So this could have been abundance, which is just the absolute count of macroinvertebrates, taxa richness, diversity, so things like Shannon's index or Simpson's index, a subset of a population, which could be any number of like a single family or a group of families or even a single species. Biomonitoring indices, so sediment-specific indices or indices for more general ecological health, and then trait-based indices, which I'll talk a bit more about later, but that essentially means quantifying a macroinvertebrate by its functional roles that it carries out and provides, as opposed to just its Latin name and its taxonomic name. And then the detectable effect was the direction of the response. So, for example, a positive effect of abundance would be an increase in abundance as a response to fine sediment. I also assessed the evidence quality of each publication, so I used the eco-evidence method, and this essentially scores articles based on their study design. So more comprehensive study designs, for example, before, after control impact studies with multiple replicates than a And then what I essentially ended up with was a series of count data. So each line of evidence represented a single count, and then this was weighted by the evidence. And the appropriate analysis for this is chi-squared. And I was looking at the associations between the response or predictors with the detectable effect and whether this differs significantly from the null distribution. So to summarise some of my results, firstly, some of my systematic mapping kind of analysis. I counted the number of publications which cite fine sediment per year, and then I standardised this by the number of papers citing a much more general, broader terms such as environmental science. And what I found was there was actually a relevant decline in papers which research around fine sediment, probably since about the mid-80s. And considering that we know now that this is a significant cause of ecological decline, it's quite concerning that we're not really focusing on researching its effects more. I also mapped the countries where each evidence came from, and this showed that there was a global imbalance of publications, and it's quite clear which countries have research groups looking at this issue. However, countries where there's no evidence, there's a lot of differences in land use management and climatic challenges, which could have some really interesting evidence for macroinvertebrate responses to fine sediment. There was no kind of globally agreed fine sediment size definition. So at the start of my presentation, I defined fine sediment as particles smaller than 2 millimetres. That wasn't completely random. That's related to the Wentworth scale for sand, silt and clay. and also there's an ISO standard for sediment size. However, we know that the ecological response is quite heavily dependent on the grain size. So things like that make comparisons between studies really difficult, and a large number of articles didn't actually quantify the grain size. These ones were generally what sampled the sediment in suspension. A lot of articles presented information on more traditional metrics, so abundance, richness and diversity indices. So the figure on the top right just shows the raw frequency counts and the direction, the detectable effect for each of these. And the chi-squared analysis for this was significant, and this bubble plot here shows the size of the residuals, which is what we can use to look at where those significant differences lie. And essentially what this shows us is that an increase in fine sediment is likely to result in an increase of abundances or density, and an increase in fine sediment is unlikely to result in an increase of richness or diversity. And this is definitely supported by what we know from the literature in high sediment environments where there's low oxygen exchange and a very homogeneous habitat. We tend to have taxa persist, which are very small in size and tend to persist in high numbers, and these dominate, having relatively low diversity of other different taxa. When I looked at articles which recorded trait responses, there was actually only enough data for 2 trait groups, which were feeding mode and mode of locomotion in relationship to substrate. Using traits in this way is a relatively emerging approach, so this could be often recorded with it. So when I carried out my chi-squared analysis on this evidence for functional feeding groups, this showed that shredders were more likely to respond negatively to fine sediment. And when we think about mode of locomotion and relationship to substrate, burrowers were more likely to respond positively. However, this relationship is likely to be quite complex, as it depends on the size fraction preferences of the organism. So some taxa like burrowing in sand size fractions, whereas others prefer really fine sands and silts and clay even. And when I was carrying out my review, I often found that there was evidence for responses, but no explanation of the mechanisms behind this. And these are just a few quotes that I found across a number of papers. And the summary was that we can see what's happening, but we don't know what is the mechanism behind this. And certainly further work is needed to try and understand exactly why this is happening. Leads me on really nicely to my second phase of my PhD, which was to try and improve the mechanistic understanding of the effects of fine sediment. So I specifically was looking at sorry, I'm just glitching here. So I just want to return to the figure that I showed near the start of my presentation, which was this, the direct and indirect effects of how suspended sediment affects community composition. And the relationship that I specifically wanted to look at was how the end in suspended foods, specifically on the physical effects, and the terms clogging and abrasion are mentioned quite a lot in the literature, but the evidence remains spurious. So just to recap, the cause would be an increase in fine sediment, and the mechanism that I'm trying to understand is abrasion and clogging to soft tissues, ultimately causing a change in community composition as the effect. So in order to do this, I exposed 3Ds of macroinvertebrates at 2 sediment concentrations plus a control, and then I used 2 different water velocities to try and see whether high or low velocity would affect potentially the abrasive forces of fine sediment. And after exposure, I removed the tracheal gills of each insect taxa. So this is one of my target species. This is a mayfly, and you can see it has these gills held outwards to the side, and this is what I removed. And in order to effect of abrasion clogging, technique, which was actually developed for using visual assessments of fine sediment on riverbeds. But in this application, I used it to assess the clogging and buildup of debris of fine sediment on the gill surfaces. And then you essentially run these through a program which can assess how much of your frame is covered within this pigment or the fine sediment, essentially. So these images just show a range of different coverage across my 3 different species. So my results, I didn't find any evidence of abrasion. I would have expected to see some kind of scratches or dents on the gill surfaces, but what I actually found was coverage or clogging, so a large buildup of fine sediment and debris on the gill surfaces as opposed to abrasion. And then when I look at the results of each specific taxa in turn, for Ecdinurus venosus, which is a mayfly, fine sediment coverage of gills significantly increased with increasing suspended sediment. And this taxa has 6 pairs of gills, and they're held laterally towards the side and in a downwards trajectory. And they actually have a plate-like gill which protects underneath a feathery gill. They do use both for respiration, but it was the plate gill that I used in my exposure experiment. And this taxa is actually relatively sediment when we look at its scoring across sediment-specific indices. And that's pretty much supported by my results here, that fine sediment coverage does increase with increase in suspended sediment. Ephemera Danica, which is another mayfly, there was no significant effect of suspended sediment or velocity on coverage. This taxa has 7 pairs of gills, and if you can see from the image, they're actually held on their back and upwards into the water current. So what they did was just became nets for fine sediment across all the different treatments. This taxa are actually burrowers, and they like to live within sandy or even silty environments. And they're generally considered quite a tolerant species of fine sediment. So buildup of fine sediment is unlikely to affect them. They don't have any mechanisms to keep fine sediment off. However, they have been shown to use their legs to clean their gills, and their gills also oscillate, which could also provide a cleaning role as well. I wanted to mention as well the role of chloride cells and osmoregulation. So these are present on mayfly gills. So mainly I've been referring to the effects of fine sediment coverage on respiration, whereas you can see from these images that the debris is kind of clogging and infilling these chloride cells, and these are really important to balance ion concentrations. There's been evidence that they can vary in number according to osmoregulatory demands. So this may be the mechanism through which these taxa are affecting sediment. To carry out a similar study, but look at taxa from natural sites with a variety of different sediment pressures and see whether the chloride cells do vary in number according to fine sediment pressure. And lastly, my Caddisfly. I found that fine sediment only significantly affected coverage when velocity was low, between low and high suspended sediment. For this taxa, the gills are very sheltered under the thorax. There was less coverage overall across all treatments. They live in quite sheltered environments, and they actually construct silk nets with their mouth parts in order to feed. They don't have chloride cells on the gills, just the mayflies that have that, and they're considered moderately sensitive to fine sediment. However, based on my results, I'm proposing that this is probably related to the feeding mechanism, as in high sediment environments, these nets can become clogged and even rupture, and the organism has to spend increased amounts of time reconstructing nets or fixing nets, and obviously it's feeding a lot less efficiently as well in high sediment environments. So how does understanding the mechanisms underlying responses to fine sediment help with monitoring approaches? So that leads me on to my final phase, which was to test the response of macroinvertebrates to different measurements of fine sediment pollution. And we know that macroinvertebrates are excellent sentinels of river quality, and this has led to the development of many different sediment-specific indices globally. So these use the ecological community as a proxy for understanding the and the two which have been developed specifically for use within England are the Combined Fine Sediment Index, referred to as the COFSI, and this is the sum of its constituent parts, the OFSI related to the organic fraction and the TOFSI related to the total quantity. And then we also have the proportion of sediment sensitive vertebrates, the development, the EPSR. And these two indices were developed in very different ways. The COFSI used a purely statistical and empirical approach, and the EPSI was based on the PSI, which was expert knowledge derived, and then the E represents its empirical optimization using real data. And you can see that this graph shows a plot of each dot represents an individual taxa and its scores across each of these indices. And what we can see is some taxa are scoring relatively sensitive on one index, yet they're quite tolerant on the other. So this is obviously a relic probably of how each of these indices were developed. And it represents an opportunity to try and understand further exactly what's going on and what else could be controlling these fine sediment scores. So my aims were to compare different methods of measuring fine sediment, specifically the two methods used in the optimization of those two key indices, test the performance of these sediment-specific indices and compare them to other more general indices, determine which environmental variables and antecedent conditions affect index performance, and hopefully provide new insights into the mechanistic basis for fine sediment biomonitoring. In order to do this, I thought it was really important to collect new independent data from these from the two articles that developed the indices. So I sampled 21 different sites in spring and autumn, which is in line with Environment Agency timings for ecological assessment. I made sure that they were relatively clean sites, so I filtered for confounding effects such as water quality, because I was really trying to isolate the effects of sediment. I made sure that they were a range of river types. So in the UK, we classify rivers based on Agroecology called RivPACs, and this essentially uses a number of different variables like width, depth, altitude, slope to classify this into a river type. However, all my sites were all lowland because evidence has shown that it's lowland areas which are more susceptible to fine sediment accumulation. To make sure that they were a range of fine sediment risks, and this was using the agricultural fine sediment risk rating developed by Mark Nora et al. Each site was within 2 kilometres of a gauging station, so I could calculate a number of hydrological indices, and then at each site I collected biological and physical data. So these maps show each of my sampling locations. On the left, they're colour-coded by RivPAC's N group, and on the right, they're colour-coded by the risk ratings. So this goes from green, which is 1, which is low risk, all the way to red, which is high risk. And then the different methods that I use to measure fine sediment. First, I used visual estimates. So these images, these show kind of what you would be expecting to do, which is estimate what percentage of sand, silt and clay persists within that area. These pictures are more carried out at the patch scale, but what you would carry this out in your whole sampling reach. And there is some guidance on this in the River Habitat Survey handbook. It's not just completely random. And then I also carried out the resuspension, also called the disturbance method. And this involves agitating the water within a set area and taking a sample. So by knowing the diameter of your cylinder, the volume of the overlying water, and then the mass in your sample, you can then calculate a mass of sediment per unit area of river bed. Carry this out to estimate surface sediment just by agitating the surface water, and then again in the top 100 millimeters of the gravel by agitating with an auger down to that depth. And this method is particularly useful because you can then carry out any number of further analysis on your sample. So I carried out loss organic fractions. I also carried out suspended sediment samples, which just involved taking a water sample when I was at site. So when I think about comparing these methods, what we have here is a correlation plot. So each number represents the pairwise correlations between each of the different metrics of fine sediment. And these are labelled from sort of top left in the middle squares all the way down to bottom right. On the lower left-hand side, we have the absolute raw data just plotted just to see patterns. And on the right-hand side is the Pearson correlation coefficients. And the asterisks represent significant pairwise correlations. So in the past, visual fine estimates of fine sediment have been criticised because they have high user variability and it can be quite subjective. However, I find a really high and significant correlation between visual estimates and total surface sediment from the fully quantitative method. And then another criticism of visual estimates is that it only assesses the surface sediment, and this could be unrelated to the fine sediment that's embedded within the top layers of the gravel. However, although the correlation coefficient was lower, this was also quite strong and still significant. Interestingly as well, sediment is very unrelated, not significantly correlated at all with any of the deposited metrics of fine sediment, which is what we would expect really, but it just highlights that when we're talking about physical and ecological effects of fine sediment, we really do need to make sure we're clarifying what fraction we are discussing. When I tested the performance of the sediment-specific indices, again, this is another correlation plot, just slightly different. The colour of the number represents the sign of the correlation coefficient. And again, asterisks represent significant pairings. We can see from the top left, we have the PSI, EPSI, and the EPSI FAM, which is just the EPSI FAM is just a mixed taxon level iteration. So it's a bit more useful in real life situations where you may not always be able to identify everything in your sample to species level. And then we have the OFSI, TOFSI and COFSI. And the section we're interested in, I've just highlighted in the blue rectangle. But the hypothesis was that the EPSI group of indices would correlate better with visual fines estimates because this is the metric that was used in its optimization. And then similarly with COFSI, I would have expected this to have a stronger relationship than EPSI with total surface quantitative estimations. The gradient was used in its optimizations. However, what I actually found was that it was the EPSI Mixed Taxon Level Scoring Index, which was highly and significantly correlated with both the visual fines estimates and total surface sediment. When we look at indices which are not sediment specific, they are more related to other pressures or more general ecological health. So from top left, we have the WHPT index. This is the Wally Hawkes Paisley Trig Index. This is actually an empirical organisation of the WP just below it, which has been used for many years in ecology auditing agency. However, it's estimated that this is perhaps more related to organic stress as opposed to general ecological stress. We then have LIFE, which is the low tick index flow evaluation. So this scores taxa based on its sensitivities to low flows. EPT, which is the richness of taxa within the orders Ephemera, Plecoptera and Trichoptera, which is your mayflies, stoneflies and caddisflies. We then have raw abundances, Shannon's diversity index, and then a couple of functional trait indices is functional richness and FDIS is functional dispersion. And then we have shredder percentage and the community weighted means of shredder, which is a callback to earlier in my PhD where I found that the wealth of evidence shows that shredders were unequivocally sensitive to fine sediment. So again, the area that we're interested in is within my blue rectangle. And the key point really is that none of these indices performed better than any of the sediment-specific indices, specifically EPSI-FAM, which was the best sediment-specific index. But interestingly, the EPT index was a good estimation of organic stress, both surface organic sediment and subsurface organic sediment. I also looked at gradient forest analysis. Now, this looks at taxonomic or trait-based turnover across your pressure gradient diversity. And what I found that taxonomic turnover plateaus before trait turnover across the pressure gradient. So despite the functional indices not really explaining anything to do with fine sediment in my previous figure, you can see that they do provide more information than using taxonomic metrics alone. And also, most turnover, both taxonomic and trait-based, occurred within the first 50% across the pressure gradient for visual fines and 25% for total surface sediment. So perhaps this indicates that we should prioritise land management interventions in areas where there is greater potential for change, so i.e. where sediment is currently low, yet has the potential to become higher over time. And these tables, I just wanted to show them quickly. These are related to the gradient forest analysis because it can pick out which taxa or which traits are contributing most to those curves. And shredders, again, were pulled out as the trait which was contributing most to the turnover for visual fines. So just, again, more evidence that shredders are sensitive to fine sediment. So to conclude, the weight of evidence points to shredders as sensitive to fine sediment and burrowers as tolerant. The experimental results showed that gill surfaces were covered in fine sediment debris to varying extents. However, this was dependent on the treatment and gill morphology and behavioural responses as key factors. Visual estimates are a relatively reliable proxy for fully quantitative resuspension methods. The PSI mixed, or FAM as I called it in the plots, was the best performing sediment-specific biomonitoring index, and trait-based approaches show promise. However, they require further development before they can be integrated in the same way. So I just wanted to quickly give an overview now of what I'm going to be spending the next year doing, and my postdoc title is Ecologically Resilient Catchments. So for the past 3 years, I've been focusing purely on fine sediment, whereas we know that aquatic environments are multi-stressor environments, and this can then be confounded by climate change and how this will affect the community in the future. So resistant ecosystems show little impact due to repeated disturbances over time. However, if disturbances become too intense, structure and function may be severely impacted to the point that the ecosystem does not easily recover or recover at all. By contrast, a resilient ecosystem is often immediately impacted by even low intensity disturbances. However, this has the capacity to quickly recover structurally and functionally to pre-disturbance conditions. So I'll be specifically looking at how we can measure this using two different approaches: the community assembly approach and modelling, which specifically looks at the processes underlying change, and then ecological stability, which hopes to quantify the dynamics that lead to change. And I've mentioned taxa and trait-based approaches before, but specifically looking at whether we can develop trait-based approaches within this context, because they do have benefits such as being able to transcend boundaries in taxonomic distributions between regions. They can avoid overemphasis or underemphasis of abundant species, and they can provide a greater mechanistic understanding of the interactions between the environment and the ecological community. So my objectives will be to identify which traits and derived metrics can be used to understand resilience, explore whether we can develop indicators of where ecological function may be compromised, and develop the concept of recovery potential in aquatic environments. And then finally, hopefully summarising this and making recommendations on the potential for the approach to be applied at a national scale. So thank you very much for listening.